It's our pleasure to host you to this scientific seminar, which is Biodiversity and Applications of Marine Microorganisms and Enzymes. This seminar is going to give very kindly from some members of the European project with H2020 funding, which is the InMare project. It's innovative screening platforms to discover and use the functional protein diversity from the sea. So we are hosting here today 24 institutions from across the European Union, they have very kindly accepted to offer this scientific seminar to you. And this seminar will also be recorded on streaming for all the public which is interested in this topic. So we have here today Professor Peter Golshin from the UK, Dr. Mikhail Yakimov from Messina, Italy, and Professor Manuel Ferrer from CSIC, Institute of Biocatalysis, and he will present the speakers and give a scientific background of the speakers. So thank you very much, Manuel.
You're welcome. Thank you very much, Anna, for this kind introduction and for giving us the opportunity to present our work and to be here. Also to the University, to the Universidad Católica de Valencia, for hosting us for the meeting, and also for the opportunity to be here, and also all of you for coming here to this seminar. So I want to introduce the three speakers. So the first one will be Peter Golshin. He's at the university. He's expert in marine in microbiology and molecular biology and marine biodiversity, and especially focused on biodegradation and on the capacity of the microbes to degrade pollutants, and in the general sense in marine biotechnology. The second speaker will be Mikhail Yakimov from the CNR, from the Institute for Biological Research and Marine Biotechnology from Messina, from Sicily, and he's one of the top experts in marine biodiversity, especially in extreme environment, and particularly in the deep sea hypersaline and oxide basins in the Mediterranean Sea. So probably you will hear this type of environments. And the last one, I'm myself, so I introduce myself. I'm Manuel Ferrer. I'm coming from the CSIC, from the Institute of Catalysis in Madrid. And as the name of my institute mentions, so I'm more in the focus of catalysis, how the enzymes that we use, either from soils or marine environments, can be to produce something which is of interest for us. And with this, I think it's a pleasure to introduce Peter, who is going to give the first presentation. And if you have
any questions, if you could please until we have the 3 short seminars, and then we will have a discussion time session. Okay? Thank you.
Thank you for this nice introduction. Manolo, and thank you for being so numerous here. I'm going to talk about which possibly we started maybe in mid-1990s, dedicated to the marine oil degrading microbes. So I'm coming from Bangor University, and it's situated just right here. So for those of you who doesn't know its location, so I just want to see what the pointer is. Yeah. So it's just there. Yeah. So this is northwest of England. This Wales is located here, and we are situated in this spot. Yeah. And it's small town and small universities of about 12,000 undergraduate students and 2,000 staff. And the city, it's small, one of the smallest city in Britain. It's about the same number of inhabitants, maybe 15,000 locals and 12,000 students. So we are going to talk about oil pollution and marine microbes that are degrading petroleum constituents. And you know that there is a huge release, accidental by intent, of petroleum in the sea. So you all are aware of those, dramatic events like, the tanker accidents and so forth. But in fact, they make only a little proportion of spills in the sea. The most massive one is natural seeps. As you can see, this is the segment. It's about 50%. Does it work? About 50% of all petroleum entering the sea is coming from natural seeps. And if you look at the statistics of tanker accidents and spills produced, starting from 1970s, you see that the security of tankers has dramatically increased. So the level of pollution of the sea due to the accidents went down very strongly. And very recent event, not very recent, but already in 2010, Deepwater Horizon, oil rig explosion and accident in Gulf of Mexico was about 700,000 tons of petroleum, resulting in this quantity of petroleum entering the sea. So you see, these are the coastal area, the most affected marine areas, the most affected by petroleum pollution. Due to the transport, we have accident spills due to the use of fuel for vessels, and also from the past, so-called ticking time bombs. We have lots of shipwrecks situated on the seafloor after the military conflict. So this is, for example, the American tanker Mississippi, sunken by Japanese kamikaze airplane, and this is how it looks now. So it's very shallow. You see that this is the leak. It's not the coral. It is the petroleum still leaking from the sea, from the hull of the vessel. So there is also another problem, is that basically there are plenty of operations when people mine the petroleum from deep areas of the sea, and it's generally cold. Yeah. So you see that when the temperature is quite low, all processes for biodegradation are very slow, and then it's a part of the problem. Deep sea oil drilling is one of the problem. Also, Arctic petroleum mining in the Arctic is also another issue. So once petroleum enters the sea, you know that it undergoes several. For example, nature of weathering is being exposed to the sunlight. Yeah, it may sink. So because petroleum is composed in myriads of compounds, millions of different chemical compounds, and so in the first instance, the light molecular weight constituents get evaporated, and some of them that are very dense, they will sink.
Excuse me.
Yeah.
Sorry.
I use this one. Okay. Good.
Yeah,
Right. Part of it is being sunken because of its density. Some fractions are quite heavy. But sooner or later, you know that oil disappears, and at least if the fraction which is not sunken is being degraded by microbes. Yeah? So this is the good news. And the rate of degradation depends on the composition of oil, because you may have very light oils and very heavy ones. So which is composed by bigger fraction of polyaromatic compounds. And it depends on basically the degradation rate will depend on the composition. So this is the very general scale of time scale of microbial response after typical oil spill in the sea. So we have low molecular weight aliphatic hydrocarbons that being either evaporated or biodegraded within maybe a couple of weeks. So there are high molecular weight aliphatic hydrocarbons that take longer to be degraded. low molecular weight aromatic hydrocarbons, they are not a big issue if you have oxygen. But there are some polar fractions and high molecular weight aromatic hydrocarbons that tend to sink in the areas where you have no oxygen, and it will take geological times for them to be degraded. So it's thousands and thousands of years. And typically, so once you spike the petroleum into the seawater, you will see the emergence of alkane degraders, aliphatic alkane degraders within a couple of weeks. And then the aromatic biodegrading microorganisms turn on after some delay. So this is the typical dynamics of growth of natural marine microbes as a response to the petroleum pollution. And as you can imagine, this is the natural compound. This is not xenobiotic, yeah, in strict sense. So they're natural compounds. So the hydrocarbons have a lot of carbon, yeah, and so they make very perfect food for microbes as a source of energy and carbon. The problem is that they are hydrophobic, yeah, so they don't mix with water, and they normally have very low oxygen content. And as you know, oxygen is the ideal electron acceptor. So all processes where we have oxygen as an electron acceptor, they run much faster in biological systems. So microbes must tolerate the impact of hydrocarbons and their membranes, because they also, hydrophobic compounds, they dissolve membranes. And they must have the hydrocarbon oxidation metabolism. So, as I mentioned, if you have oxygenated seawater, all processes run much faster than without. So, since a while, so since 1995, so when we, myself and Mikhail Yakimov, who is going to talk about something else today for a change, we were starting isolating real marine hydrocarbonoclastic microbes. Hydrocarbonoclastic means hydrocarbon degrading. Anecdotally, before that, people were dealing with typical, soil microbes like Rhodococcus, Pseudomonas, and so forth. So, but those were, the first one was Alcanivorix borkumensis, isolated from the sediment of Isle of Borkum. It's on the German-Dutch border, after, getting an enrichment culture of petrol, crude oil added to the sediment and seawater. So it was a new genus and species, and apparently it's a new family, Alcanivoraceae. So then we have isolated Thalassolitus. It's from a heavily polluted area, from a petroleum terminal in Milazzo. It's in Sicily, North Sicily. Oleophilus messinensis, as you can see. So I was personally taking the sediment from Messina Harbor, which is heavily exposed to the naval traffic. So you have a lot of ferries going to the mainland and to Sicily. And it's another new genus and family. And this one is these are 4 examples of the microbes we have isolated. Misha brought from Antarctic expedition some petroleum-enriched carbon, which we then later described as Oleospira antarctica. So the interesting thing about these microbes, they don't really see many substrates. So if you feed them with amino acids or sugars, they will not grow. So they prefer petroleum-like compounds like fatty acids, alkanes, or alcohols, long fatty acid chain alcohols. So this is what makes them very different from the rest of microbes. And they are naturally occurring in the marine environment, which is also very important. So we have few genera of oil degraders. So those on the previous slide, they are, let's say, they have restricted metabolism. So they're eating almost exclusively alkanes and similar compounds. Whereas the first one, which was isolated much longer before, was Marinobacter hydrocarbonoclasticus, which is more versatile. Yeah? So it was isolated by a French group back in 80s possibly. And it was then called Pseudomonas nautica, but now it's was renamed later after maybe 1990s, Marinobacter hydrocarbonoclastic. It's very versatile. It's also able to grow at the expenses of nitrate respiration, so in anaerobic conditions. This is about phylogeny. Most of them belong to the gamma proteobacteria. It's quite abundant marine group. So, and this is their specializations: Alcanivorax, Thalassolitus, Oleophilus, Oleospira, Cycloclasticus. Is Cycloclasticus and Neptunomonas. They're specialized in hydrocarbon and polyaromatic hydrocarbon degradation. So, whereas these four guys, they prefer alkanes and linear and branched. So you see this quite old map showing the ubiquity of all those hydrocarbonoclastic microorganisms. So they naturally occur in the environment at different depths, or in the environment that resemble marine environment with high salt concentration and a presence of petroleum. So we'll be asking how they exist naturally without input of petroleum. So this is the answer. So some of these microbes are associated with unicellular primary producing eukaryotes like microalgae that, as you can imagine, they produce similar compounds like fats, fatty alcohols, and similar molecules. So, and some of these microbes are not able to propagate from their resting cells without Alcanivorax, for example. This particular case was described by Chris Bulk from University of Tasmania. So this is the natural carrier of these hydrocarbon degraders, because they cannot sit and wait for a tanker to collapse. Yeah? So they feed on natural substrates. Okay, so Alcanivorax was the first, and we were able to characterize that by sequencing his genome maybe 13 years ago already, and we did all these omics studies on proteome, response of this microbe to the shock load of alkanes. And what we have established is it has a reduced number of metabolic pathways, streamlined genome, increased number of alkane hydroxylases and monooxygenases and cytochrome P450s. So these are the enzymes that catalyze the first reaction in alkane oxidation. It's terminal oxidation of alkanes. It produces high number of transporter proteins for nutrients, and it produces very effective surfactants. So the thing is that this Alcanivorax has also been found by our Japanese colleagues in the Sea of Japan after there was some tanker accident. And they say that they first showed that actually the population of these hydrocarbonoclastic organisms was very high. They made up to 90% of total population just within a few days after pollution. Yeah? So it's general feature of these microbes. They bloom and flourish after they see the petroleum. And so this high number of transporter proteins for nutrients enables them to grow. Essentially, they outcompete the other microbes by taking up the mineral nutrients. As you can imagine, so for a balanced growth, you need the proper composition in your medium of carbon, nitrogen, phosphorus, which more or less resembles the gross formula of microbial biomass, which is pretty constant. So if you have 50 molecules of 50 atoms of carbon, you need to have 14 of nitrogen. And in the sea, nitrogen is very depleted. Nitrogen and phosphorus. Once you have the petroleum, which is insoluble, you have an excess of carbon. So you need actually to provide the nutrients, yeah, for an effective growth of these microbes, and they're able to acquire these elements. So what we have seen also, so all these clusters for alkane hydroxylation, they are situated near to the origin of replication in the genome, which actually makes higher gene copy dosage. So you can imagine when the replication forks reach the terminals here, you have many, like 2-3 more copies of genes in this area than in that. So this is one of the mechanisms these microbes are, using to increase the number of gene copies, and therefore the quantity of protein relevant to degradation. So what is, I'm going to chat about a little bit about the cold temperature as the limiting factor and specific group of microbes that are thriving in the cold. Yeah. So as we mentioned, oil plumes in the deep sea, World War II wrecks and deep sea oil drilling are happening at very low temperature. As you can see here on this graph, below, let's say, a couple of 100 meters, the so-called thermocline, and temperature decreases from the surface, which can be in tropical area about 25 degrees, down to almost the freezing point, down to 4 degrees. Yeah. So, and it's a big constraint for the degradation rates. So, and this what was observed actually in this Deepwater Horizon accident in 2010. So there was a huge spill of about 7 million tons of oil released, and a very small fraction of that was dispersed. So there was a huge plume of oil formed at the depths of 1,200 meters. And so what people found, there was a paper in Science back then that this is the group of microbes that have been sitting there and possibly degrading the petroleum. And so among those was this Oleospira antarctica, which I mentioned, which we already had in the lab since 2001, possibly. And this is called adapted oil degrader. So we did genome sequencing, and we have found that this color corresponds to the number of gene clusters for alkane oxidation. We have found it that alkane monoxygenases are overrepresented there, and ALK-B, just one copy. But these are, so what you can see here. So this is Oleospira, this is Alkanivorax, and this is Marinobacter. So all those microbes, they have enhanced number of gene clusters for alkane degradations. For example, this is how it looks like on the very schematic view. ALK-B is alkane monooxygenase, non-membrane. This is P450 cytochrome, which acts in the concept of ferredoxin reductase. And both of these enzymes, they actually hydroxylate the alkane, as you can see here. It's hydroxy group. And then you have alcohol monooxygenase, aldehyde dehydrogenase, and so on. And then finally, this fatty acid end up in the beta oxidation pathway. So these organisms have enhanced number of gene clusters. So if you look at the common views on the cold adaptation in marine microbes, in microbes generally, so there are a few mechanisms we know, like fatty acid desaturation at low temperature to increase the fluidity of membranes, production of solutes, amino acid composition of bias in proteins for sacrophiles, and production of a suite of proteins for cold adaptation and cold stress, like chaperones and protein folding machineries. So it was the same case in this Oleospira. So we have increased the saturation rate here of, this is the saturation, yeah, this is fully saturated fatty acids, depending on the substrate, yeah, so you have slightly difference. And it works also there in this microbe. And they also produce solutes. So what you can see here, the red ones is Oleospira, this is Hachella, this is Marinomonas. So you have the color codes is 3, 2, 1. So the more if you have complete pathway, all of these boxes are colored. So it's ectoine synthesis and it's betaine. So only 15 of 343 at the time gamma protobacteria have both clusters. So this cold adapted microbes has both mechanism to increase its tolerance to the cold. So this map shows what's going on in the cell, which part of the proteins are interacting with chaperonin, which is the main protein folding machine. So you see here, absolutely different picture here than from plus 16 degrees. 16 degrees is more stressful, but the microbe grows much faster. And you see lots of pathways related with the peroxide, so how the microbe deal with peroxide, you know, there's glutathione peroxide, peroxidases and so forth. So whereas here, most of the chaperone substrate are related with pilus formation, for example, or detection of TONB dependent receptor, this for scamagic proteins, and some of the central pathways of central metabolism. And from this project, we were lucky enough to be able to establish E. coli, which use chaperones from these cold-adapted microbes. And the company has, this company at the time, strategy now is Agilent. So it sells the E. coli with so-called Arctic Express competent cells that are maybe helpful to you to express your proteins if you are desperate at some point in time. So because some people cannot express, so some proteins are not very well expressed, and you try different things. So this particular system was efficient at least in 400 to 500 cases, as we can judge from Google Scholar references. So is amino acid compositional bias relevant? We show that not. This is Alcanivorous borkumen, this is Olei spira, and this is Bermanella maris rubri. So we have very congruent distribution of amino acids in peptides for Arctic or deep sea microbe and in Red Sea microbe. Yeah. So we also milk this genome for proteins. So we have cloned about 200 proteins. And surprisingly, many of those were not called active. So some of them were, but most of them were like adapted to higher temperatures. And we have crystallized 12 or resolved 12 crystal structures within this project, which has more than double the number of cold-adapted proteins in the PDB. And we found that general distribution of charge is, tends to be more negative if you compare Olei spira proteins to the mesophilic counterparts. So just one slide per each, and we finish this. So there are also, apart from bioremediation and natural oil degradation, these microbes can have good application in terms of production of biosurfactants, enzymes, bioplastics, and compatible solutes. So this is the example of biosurfactant from Alcanivorax. This one is a glucose lipid. Yeah? So you know that it's like biological soap. To increase the to solubilize petroleum or oleophilic compounds, you need to apply some surfactant, and it increases the surface of contact of microbes with the substrate. And there is a number of lipids, rhamnolipids, sulfurolipids from different resources, not necessarily from alkane degrading, but alkane degrader have also great potential for those. Enzymes, this is what Manolo will be talking about, and this is what we are after in Mare project. So you know that enzymes can catalyze many reactions, and they do that very specifically at times. And this is the list of enzymes that are of industrial interest. You know, monooxygenases, hydroxylases, esterase lipases, dehalogenases, and so forth. And also, this is non-enzymatic protein, which is of industrial relevance as well, which I just showed you. So this picture indicates this is actually our mutant of Alcanivorax, which we spontaneously got. This is the cover of Journal of Bacteriology, and it shows Alcanivorax, which produces extracellular polyhydroxyalkanoic acid, so biopolymer. Yeah? So as you can imagine, the main storage compound is polyhydroxybutyrate and similar compounds. Sometimes it's copolymer of a few different ones. And as you know, so in coming few months, the single-use plastic will be banned from use by the European Commission. And so this is what we're looking after, so biopolymers. And these microbes have great potential in that respect. Solutes, as I mentioned, compatible solutes to stabilize the proteins and stabilize enzymatic reactions with a range of application, medicine, enzyme production, biocatalysis, and cell culture. And so I would like to stop here. There are a few conclusions. So we have a very interesting group of marine microbes. So they are very capable to degrade, and they're always present in the environment. So they start, only a limited number of them become predominant after the oil spill. So they use hydrocarbons as single or one of the rare sources of carbon and energy. So the degradation rates depends on temperature, supplement of minerals and oxygen. And to survive the competition, they specialize in certain substrates that are adapted to different temperatures. You have some niche specificity among these microbes. And we also have further applications biotechnology that may still come from this group of microbes. And I would like to thank the funders in the past and our colleagues, in particular from this INMARE project. Most of them are occupying the rear row in this room, and many of those were involved into this research. Okay, thank you very much.
Someone will help me. Good. I'm second who will present the talk, and as you see, the title of my presentation is a little bit ambiguous, but I like very much this actually topic. And I'm Mikhail Yakimov from CNR. Now we have the new name of institute, which is Institute for Biological Research and Marine Biotechnology. And Messina, we are located here. And actually, we are used in last 10 years going to this region, which is actually called Mediterranean Ridge. And this area harbors very interesting, peculiar geological formations, which called deep sea anoxic brine lakes. And actually, last finding, especially, I think this one, it was the recent paper. Oops. Sorry. Recent paper in Science by Italian team who actually documented or somehow almost confirmed the presence on the south pole of Mars some subglacial lake. Subglacial, it means we have some thickness of the ice above 1,200 meters, and there they documented presence of the liquid water. And actually, why I said about the peculiar formation of the anoxic lakes, this we have at least now we have nine formations, which are really deep sea lakes. You can see when you go with the ROV, you can see there nice pictures like this one. You can see very nice beaches. And then lake is coming here. Here, for example, another photo made by ROV Jason. And yeah, you can really see the situation. We have the normal sediments here. And then you have deep sea lake, which is saturated by different kind of salts. And actually, I go back to the slide to explain you a little bit in details what was happened in Mediterranean in the past. We have this, it's called last salinity crisis in Mycenaean epoch, and we're speaking about something like 6 million years old, when the Mediterranean get actually dried, and it's still discussed. It was marginal basins, that means dried just in some places, or basin-wide. Then all Mediterranean was dried off. And it seems to be then recent theories, they actually accepted both scenarios. Then we have sometimes, because it was like a temporary drying off and then flooding again the Mediterranean Sea, and we have in some places in Mediterranean, we have the sediments which they have thickness around 3,000 meters or something like this. It's really amazing amount of salt accumulated under the sea, on the bottom and the subsurface sediments. And actually, I told you that Mediterranean Ridge, it's some place where the tectonical platform, African and Asian, and Eurasian, they are coming together, and the movement of African platform caused very active tectonic activity there, and we have really the fractionation, we have a lot of volcanoes, and we are speaking about 4,000, 3,500 meters deep. It's really interesting, peculiar area, and exactly there, these lakes are located, and actually origin of them mainly depends of the two kind of, okay, these are, two kind of scenarios. For example, first one, when this dried or this sediment layer became in contact with seawater, and in other scenario, we have then within the sediments, we have already the lakes, which are ready like a lens with the brine, which are ready to be exposed and to occupy some close depression, close to the event when the brine exposed and occupied this niche, forming the lake. The lake is really a lake because they are not mixing with seawater due to the elevated density. Okay, here they are, and here I will present you in red. You have the lakes which are saturated with magnesium chloride, and it's saturation, we have something around 5 molar, even in some cases, for example, discover it's above 5 molar of magnesium chloride. Just make calculation, we have salinity over 600 gram per liter, not per kilo, because these lakes are very dense, and density is approaching 1.23, 1.25. This means it's very dense, and peculiar stuff of these lakes, they are stable due to very deep location, whereas the current minimum. This means we have lakes which are somehow can be analogous of this subglacial lake on Mars, because we we already evidence the presence of different kind of salt in Mars, which can be magnesium chloride or magnesium perchlorate derived. This means that actually some kind of analogous we have. And I don't know, from internet I found the phrase, if you want to study aliens, stay home. And we start to study aliens there, because if you will keep in mind, then we have these multiple extremes, deep pressure, absolutely aggressive environment, which is not marine and not derived from other kind of environment typical for Earth, like this magnesium saturated lakes. Then maybe only one parameter which is still missing is the low temperature, because this lake on Mars can have temperature below or around minus 70 degrees. In our case, we have plus 16, but fortunately, we have recent articles of one Australian researcher, Riccardo Caviccioli, who was studying Antarctic hypersaline lakes at the temperature minus 18, minus 20, and he actually documented the metabolic active cells there. This means it's already almost we covered or we made tick on all factors which can be somehow analogous for the extraterrestrial ambience. And this is new lake we discovered recently, a couple of years ago. We call this Hephaestus, as you can see. Yeah, this is just recent publication. You can see the 2019. In Mare was a knowledge. And as you can see, this lake, it seems to be the youngest Atlas-Sahaline formation on our Earth. Youngest, what does it mean? We just made some analysis. Oh, okay. Analysis of the diffusion of major ions. And you can see the model which we applied here. This is the points which actually experimentally proven findings of decrease in concentration of chloride and magnesium. Here we have stable isotopes. And when you apply the model of 700 years, we are perfectly fitting inside. And actually, what does it mean? We have formation which is very young, but this formation is plenty of very strange life. Okay. This is another verification that the lake is very, very young. For example, this Krios basin, it's somehow, we're speaking around 2,000, 3,000 years old. And you can see here it's profiling of the organic. And here just you have the axis, which is hydrogen carbon, and here oxygen carbon. And you can see a lot of oxidized carbon substances we have in Krios brine. But in Hephaestus, there are none, or almost none. What does it mean? We have open system, which is actually, we have then the seawater column, which is 3.5 kilometer, because it's very deep. And actually, the contact with seawater, with oxygenized organic carbon should occur. But yeah, it somehow is, but very few compared to the more older basin. That's another improvement, then we have the very young formation. This lake actually, and when we studied really the presence of real life in the brine, I would say, unfortunately, all these lakes are dead. They are sterile. Sterile just because we have two factors of magnesium chloride which are negatively affected to living forms. First one, it's chronotropic. It's really starting to, or trying to somehow organize the biomolecules, degrading or denaturating them, or in case of DNA, it's stabilizing the duplex and cannot be replicated in the high concentration of magnesium. And at the same time, magnesium chloride at elevated concentration denaturate the proteins. Denaturate, this means making them not active, but not degrading. That means we have, for example, Peter was a witness, we found many absolutely intact organisms in the brine of the lakes, these magnesium saturated, or in sediments, but all of them, they were just mummies, mummified. They were dead for I don't know how many years, absolutely intact, perfectly preserved. And the peculiar stuff of these lakes, and maybe eventual ecosystems on extraterrestrial ecosystems on Mars as well, the interface is alive. The interface in case of, for example, Krios, you can see here the photo, it's actually not from Krios, but from another deep sea brine from Red Sea. You can see here it's some kind of hot top or diversity richest place. This is the old stuff only alive. And actually we have life here. I made the arrows where we are approaching the edge of life. This one is alive. Below, no. And this layer is inhabited by many interesting, peculiar microorganisms, which we called in our first initial articles as MSBL candidate divisions, which is Mediterranean Sea brine lakes candidate divisions. And actually, at the beginning, it was some kind of, oops, yeah, it was more than 12. Now some of them renamed in different other names, but most of them remained as MSBL candidate division, and I will speak about one peculiar one. It's this one. It's some kind of archaea, halophilic. Okay, what we have? Here I put, it was the famous article of Harrison in 2013, when he put all cultivated microbes in the multiple extreme, let me say, 3D map. And here I put the point where actually we're facing environmental condition of deep sea lakes. And you can see we are far away from any cultivated organisms. This slide I prepared something like 3 years ago. Now we changed a little bit this stuff. And what I have to point, to highlight on this slide, then all these interfaces, they are extremely profitable for microbial life, for all kind of energetic reactions which can use different donors and acceptors and provide or actually live with this kind of metabolisms. And we have fermentation, sulfur or sulfate reduction, and methano and acetogenesis. All of this stuff, all of these reactions actually energetically feasible, even we are speaking about extreme salinity in these lakes. Here again, how you can actually study them. First, when I show you on the present slide, you can somehow preview the type of metabolisms which is occurring there. And then you can go and actually monitor the disturbance or the stratification of these organisms using different key functional genes for any kind of metabolism. In this case, you can see, for example, this one is ammonium oxidation, which is driven by taumarchaea, and actually seawater dominated deep sea water, at least here, the process to actually to fix ammonia and to proceed with this reaction through the whole water column, seawater column, but as you can see, it does not penetrate too deep in the interface of the lake. It stops somewhere here because all of the stuff is made by mRNA, which is actually only one molecule which is stable in these conditions, because I forgot to say you cannot study the life here using normal approaches, isolate DNA. What? It's from the mummies, it's dead. Isolate ribosomal RNA. Even with ribosomal RNA, we have the same problem. It's very stable in magnesium chloride saturated conditions. You cannot use it as a biomarker for the life. You can go only with the mRNA, which is the actually less stable among all these molecules which you can use for the biomarkers. And you can see here, for example, this is the methanogenesis. You can see here penetration a little bit deeper. Then you have here it's one specific gene which we was using for acetogenic or monitoring acetogenic bacterium of KB1 division, which first was isolated from the Red Sea anoxic lake, isolated, no, visualized by 16S RNA monitoring. And then we have here sulfate reduction. You can see here the deepest metabolism here, which you can expect. It's acetogenesis, methanogenesis, and actually sulfate reduction. And yeah, going deeper in this topic, I think I know I still have time. Okay, going deeper in this topic, we were able to at least not cultivate forever, but at least have in a couple of months and able to study the metabolism of two dominant group of microorganisms which are living in interface of anoxic lakes, in this interface lake Creus. And you can see all of them, they are really dependent on the overlaying layers as a supply. In this case, for example, supply of osmolyte is glycine betaine, which can be afterwards degraded by KB1, producing trimethylamine, which then can be degraded by this MSBL1 archaea to produce methane. And in this case, we have the process, which I will point your attention. We are producing here a lot of acetate. Going ahead, acetate story. We were also interesting, for example, we found here halobacteria on the tree, which is active because all this tree were constructed by 6NS, by ribosomal RNA, and most probably they were active in all these interface layers. And we tried to isolate some organisms which can use acetate in these conditions. And actually we were lucky because when you see the cardfish monitoring, it's specific for the peculiar taxon specific this visualization. You can see only the organisms which you want to see. And in this case, it was halobacteria specific cardfish, and we found them alive in the interface. When we make 16S RNA, fortunately, these organisms are actually present in many places on our planet, but all of them hypersaline anoxic sediments and terrestrial lakes as well. That means this group is really ubiquitous. And what is interesting, it's haloarchaea, which was actually first, or at least all this family haloarchaea before our publication. It was generally stated that all of them aerobic. And we have, we were able to isolate obligate anaerobic haloarchaea, which are really highly specialized to oxidize acetate with sulfur. This means this kind of respiration was never demonstrated for archaea, and especially for hypersaline environments, because the reaction actually is very energetically unpleasant. But they are happy with this metabolism. Then we made some genome analysis and all the general actually understanding of biochemistry. We have this very peculiar activation of acetate in two different mode. It's like with classical acetyl-CoA synthase, but we have also succinyl acetate-CoA transferases used by this organism. And then it has very peculiar respiration chain. This means we have only two respiration complexes, complex 1, complex 2, and then we have only one terminal oxidase, which is polysulfide reductase. And in this case, the respiration chain, it somehow explains why they are obligate anaerobes. They don't have complex 3, complex 4. Oh, okay, sorry. There is a second example which can use hydrogen and formate, and again sulfur as acceptor, and again new type of respiration, and again it can be found in interfaces, making the trophic network complex. Okay, this is in details the respiration chains. Also, we check this stuff, and they really, all these are two organisms, they don't have complex 3, complex 4, but they do have completely different terminal oxidases. In this case, we have the polysulfide reductase. Then we have the DMSO reductase, thiosulfate reductase, and then another one reductase, which is actually belong to different families, still unknown. And here we are with a little bit more complex situation, how actually the interface is working. And again, we have these two groups which produce TMA. We have methanonatron archaea, and then we have our players, which consume hydrogen formate acetate, and using the sulfur, which is just sedimenting from the over or upper interface. Yeah, that's all. Thank you. here just question. You can see the 3 people working. Here's 2 professors and 1 PhD student. Where's the PhD student? Okay, thank you.
To everyone who is here in the room, and also those who are looking us through streaming. So it is a pleasure to be here. And I'm going to discuss, I'm going to change the topic. We heard about the microbes, some knowledge about diversity and extreme and non-extreme, and I'm going to talk about enzymes, how we can screen enzymes from microbes, because they are important from a biotechnological point of view. So first of all, I would like to acknowledge the MARI consortium, which is represented by most of the partners here. The Universidad Católica de Valencia, as I mentioned before, because they gave us the opportunity, to arrange also the meeting and to arrange this meeting. To the European Commission, obviously, because the funding. And also to the people from my group, which is here. We are continuously searching for PhD students and students which are interested in marine diversity or diversity in general and enzymes. So if any of you is interested to join the group, so we are continually searching for those. So you can contact me or through Anna and Amanda, who is are here in the audience. So I'm going to discuss about enzymes, and before this, I wanted to share with you some key dates that I found relevant in the topic of microbiology and biotechnology. And the first one is related to microbiology. So Carl Woese in the last century already introduced the concept of microbial diversity, or diversity in general. And it was much later, 50 years later, when a gene through which we can make a universal phylogeny, so was possible. A few years later, a microbiologist established the basis for isolation of DNA, and this was done just because this guy pointed the attention that not all the microbes in a soil or in a seawater sample can be cultivated. So most of them we are not going to be able to cultivate, probably because we cannot find the condition or because we don't have enough time to cultivate. And this was the origin of the metagenomics. So isolation of DNA from an environmental sample, independently whether the microbe can be cultivated or not. We just care about the DNA sequence and the sequences we get from this DNA. And then in the last century, in 86, the DNA sequencing was robotized, was automated in the frame of this human project. And then this gives us the chance to sequence not only genomes from pure isolate, but also the metagenomes. So in relation to biotechnology, so we just heard that biotechnology is here since we started to produce beer, bread, and so on. But the term biotechnology was just introduced during the first war. And the first biotech company was created just 40 years ago. It's not really a really long time ago. It's just 40 years ago. And it was made just to produce insulin. Okay. What happened biotechnology not related to microbes, but related to enzymes. Well, so just at the beginning of the last century, the concept and nature of the protein itself was introduced. Then there was some introduction of what enzyme was doing, but it was in the 70th of the last century when the production of enzymes from a company, a Danish company called Novo Nordisk, now Novozymes, introduced enzymes into the detergents. Okay, and then we recognized that if you add an enzyme to the detergent, the clothes could be cleaner. And then in 2001, the first metagenomic business company was introduced. There was a company where the people thought that going around the world, the planet, take samples, and then isolate microbes and enzymes from this environmental sample could be of interest. Okay, and since that time, we now recognize that about 1 to 5% of the products that are in this room or in any other place of the planet are produced by enzymes or microbes. Imagine prebiotics, imagine the baby food that are pre-processed with proteases just in order the baby digests properly the food, to produce detergent, isotonic drinks, which are produced by enzymatic conversion, most of the dietetic product, and or the milk without lactose, insecticide, medicaments, and so on. But the question is, this is still too little. And the question is, the big oil, we know that they're in this petroleum area. We know that the petroleum at some point will decrease, and then we need to change to bioconversion, because the population still is growing. So we need to, from an environmental point of view, we need to change this big dependence to bio-based product and biotechnology in general. Okay, so which is the problem? The problem is actually most of the conversion in industry are made chemically, and it is expected that in the next 15 or 20 years, at least 40% of all chemical synthesis process should be replaced by enzymatic or microbiological process. Okay, because they require less solvent, they are environmentally friendly, and actually the amount of money invested in enzymes and microbes in biotechnology is quite high. But the question is, if we need to introduce more biotechnology in the future, we just need more microbes and more enzymes, because the ones that we have actually are not fitting to the process condition. And actually this will give a demand. So it is expected that the demand of new enzymes and microbes will increase, is increasing, and will increase about 10% every year. Because if we want to make more process based on enzymes, we just need more enzymes, okay? And the question is, where do these novel enzymes come from? Because the ones that we have actually in the catalog so are not really well performing, okay? And the source will be the microbial world, okay? It's a huge amount of microbes that are in our planet, and the majority of them we will not be able to cultivate, just simply because, as I mentioned before, we don't know the condition. It will take a lot of time to cultivate. So how many microbes are in our planet? So it is expected, this is just rough estimation, about 9 million microbial species globally in the planet. About 2.2 are marine microbes, and about 1.3, about this average number, about more than 1 million microbial marine species. Okay, so how many species we know actually? So this is a huge amount of numbers, but we have isolated and cataloged about 13,000 species, and every year we describe about 800 new microbes. Okay, so it will take about 1,000 years in order to isolate and characterize all the remaining species in the planet. So this is just too time consuming. So we need to change from cultivation to metagenomic tools, where we don't need to isolate microbes, which is important per se also, but to change to a DNA analysis. And another thing is not only the microbes that we know and those that remain to be known, but also what we know from an enzyme point of view. So this is a tree which showed about 9,000 microorganisms with genome available. Okay, if I show here the ones from where enzymes have been isolated, which are here in this gray color, we see that there are a lot of gaps. So the majority of microbes that have been isolated and with genome sequence available, still we don't know the potential of their enzymes, just simply because it's really time-consuming, the cloning, expression, characterization, and so on. So we need to invest a lot of time. And this is where metagenomic enters, okay? And then I don't know, I wanted to introduce just this movie. I don't know if any of you have seen this Inferno. Yeah, so I don't know whether you remember at the end of the movie where this Tom Ham is with a plastic bag, expected to be some virus inside. And then Tom Ham mentioned, to really know what is in there, we need to make the metagenomic sequencing. Okay, so this, as far as I know, this is the first movie where I have seen that they mention the word metagenomic per se. So it's something that you don't know what is in there. It's something like a black box, and you need to sequence the DNA. So then you can go for sampling, you can isolate the DNA, you can sequence the DNA, do a lot of bioinformatic analysis to see what the gene is doing, to predict functions and so on. And then you can clone the DNA, express the genes, and then check the activity by a lot of colorimetric tests and so on. And then finally, you can get to a lot of expression, cloning, purification of protein, a lot of biochemical work, okay? But the most important thing is that we can do everything starting just from the DNA, okay? Not from a culture. So actually we can take a ship or a boat or a cruise and then go sampling and then take a liter of seawater, filtrate, isolate the DNA and do a lot. So it seems to be simple, but the question is, these are the place where a lot of metagenomics studies has been done, but you see that there are a lot of studies, but not all the planet has been sampled. So it seems to be that we will never be able to explore the entire planet. Just simply a cruise costs 25,000 euro per day. So we are more or less, I mean, the budget of the cruise is a limitation per se, not only the scientific advance, okay? So the technological capabilities for bioprospecting the planet exist, but we are limited to the wide microbial diversity and the capacities to find the best microbe or enzyme that fit to the process. So this is, for me, one of the key issues. Not to find a microbe or an enzyme, but to find those that are of interest from a biotechnological point of view. And I wanted to show you the problem of this diversity and the problem of finding the best microbe and enzyme. I want to show two nice experiments that I like very much. And the first one is a really old one from Rene and Travisano. They put Pseudomonas, and then they make in a shaker, they agitate, and then they found that only one type of colonies develop. But if they put in a plate where there is no selection, no pressure at all, 3 different types of colonies appear. So it seems to be that the absence of selection per se promotes diversity, promoting poorly adapted phenotype to persist. So imagine that you don't have only one colony or one microbe, but thousands, and few non-adaptive phenotype develop. So perhaps the one which is of biotechnological interest is this one and not these 2. So it seems to be that together with the microbe you are interested, so you will have a lot of that are not really good. And then we need to distinguish those. And the other example is this. In a experiment from 6 years ago, they cultivate different bacteria, and then they put together and to see what happened. And what they see is that the adaptation of one microbial species affects the growth of the other one. So the bacteria A affect the growth of the bacteria B, but not opposite, and like this. So this means that, speaking from an environmental point of view and biodiversity, so So all microbes seems to be that are in everywhere. But if a new microbe comes to a new place, so the capacity that this microbe will have to develop in this environment will depend of the presence of the other one. So it seems to be, but that the diversity is quite complicated, and we need to find a way how to find the best one. So this, because all of the reason, what they make is that the species are not equally distributed in all environmental samples. You will have species that are highly abundant and those that are rare. So whether the microbes or the enzyme we are looking for is within this, so within this is something that we need to study, okay? The majority of the cases, those that are looking for are in this area. So it's take more technological problems, okay? And the other thing I wanted to share with you is it's difficult to isolate microbes, but also difficult to work with enzyme, because sequencing is so cheap that now we can just sequence the DNA in weeks' time, and then the cost is really much, and then the number of enzymes, imagine that we sequence an environmental sample, we get millions of genes, but most of us are not investing time to clone and characterize all of these genes. So in the past, the majority, when sequence was not available, so the majority of the genes were characterized, okay? So you deposit a gene sequence, and then you characterize. But now it's almost 0%. So the databases are full of sequences that nobody knows what they are doing. So we need to invest a lot of time to characterize what these genes encode for us, okay? So why the people that does not invest too much time in characterizing enzyme or the drugs? And it's a question of time, pain, and cost. Just to develop a drug for oncology requires about 18 years with about 1,000 million euro. And to get one drug, we need to start with about 10,000 per candidate. Okay, so you see that the investment is really, really high, and only 2 out of 5 molecules recover the cost. If we see the enzyme, how long it takes to isolate the new enzyme? So you get to the garden, you take the sample, and then you sequence the DNA, you clone, and so on. So actually, it takes about 7 years. Okay, the cost will be around 100,000 euro, and to get 10 enzymes of interest for industry, you need to pre-screen about 100,000 or even million. So it seems to be that in the way you are losing a lot of candidates just because they are not stable, they don't fit to the process, and so on. So it takes some time. This is just a real example of a company. Imagine that any of you is interested in finding an enzyme. So this particular company, Biocatalyst, can find the enzyme for you. So how long do the company tell you how long it will take the process? So the company mentioned that it will take 1 week just to think about the process. 12 weeks to develop an enzyme process, okay? Then to develop the enzyme per se, to express and so on, and then 30 weeks for the development of the process, okay? But here there is some question marks. This is the enzyme development or the enzyme discovery. So they don't promise how long they will take to discover your enzyme, simply because it's not an easy task, okay? So it will be from a year to 2 years to 3 years. And this is what we were doing in InMare, just to reduce this time frame. So what InMare is, or what we are doing in the frame of InMare. So we are looking at enzymes that are promiscuous. What means promiscuous? So promiscuous means that they can do a lot of conversion at the same time. It's an enzyme that is a type of enzyme that has a wide active site, so it can accommodate a lot of substrate. So imagine that the company wants to use the very same enzyme for different processes. So in this case, they can find a promiscuous enzyme, and then just spending just a cost to produce only one enzyme, they can use for different processes, okay? And then we are interested in marine promiscuous enzyme, just simply because, as Peter and Misha already discussed, it's full of environmental sites with different salinities, different pressure, different salt concentration, and so on. So we are sure that the enzyme from marine microorganisms will be the best adapted to industrial conditions, where we need solvent and we need high temperature sometimes, and so on. Okay, and also not only for this particular reason we are looking for enzyme from marine origin, but also because the diversity in the marine environment is much higher than in the soil or in this room, just because the diversity of microbes is higher, and then this means higher chemical diversity. So it seems to be that if we screen in a sample which has a higher diversity, so we will be able to find more enzyme. And the question is, and I'd like to photo just to introduce that if we find an enzyme and a microbe, we cannot apply only one technique. We need to apply a lot of techniques together that can be applied to identify all the genes and then find the correct one. And this is why, in the frame of European Commission funded project, is 2020, so the MARE project come up, which related to the discovery and use of functional protein diversity from the sea. I'm going just to produce, to give a summary. The idea was to produce at least one of the largest collection of marine enzymes worldwide. It's consisting actually about 1,000 protein. We do a lot of things: cultivating microbes, genome sequencing, bioinformatic analysis to predict the function of these genes. We make a lot of bioinformatic prediction. We make a lot of activity screening in the clone libraries or in the microbes. We invest a lot of effort in expression and purification. So the problem of a company now, it seems to be that it's not to find an enzyme, but to express the enzyme, because the cost in the production process is more related to the cheapest production of enzyme and the stability of the enzyme, but not only in the searching. We do a lot of biochemical characterization, and we study evolution. And why is important the biochemical characterization? Because if we characterize all the enzymes and we associated a function to a sequence, then we can make prediction. So next time, if we found in the database a sequence, we can predict whether this sequence will encode an enzyme which is of interest for us. So for the reason, it's good to invest a lot of time for characterization. Okay, so these are within the MARE project all the sites from where we isolate enzyme. So it seems to be a high diversity of sites. And the good thing is the majority, because this diversity of sites, we found that the majority of the enzymes belong to microbes we have no clear phylogenetic affiliation. So that means that they're entirely new. This will position these enzymes in the future bioeconomy. So these are just few examples of the microbes and sites, as Misha already discussed, from where we isolate the enzymes. And this is one of the things we are doing together in the MARE consortium is we have hundreds of different enzymes here. I think we have hundreds of different enzymes. So if the enzymes, you isolate this promiscuous, so you will have this. So we are finding, or we are interested in this kind of enzyme, who are converting everything. Everything you saw is they are converted. So this is what we are doing. And one important thing within MARE, so we can see how abundant are the different promiscuous enzymes that we search. And then you see here 7 types of enzyme, from more promiscuous to less promiscuous. And you see here that these promiscuous enzymes are really abundant here, also here, but not here. So it seems to be that not all phenotypes are equally distributed in the planet. So it's good to think about the strategy of sampling. It's not like you take a cruise and then you sample in this area because nobody searched before. You need to invest a few weeks or months just to find the correct sampling strategy, okay? And then once within the MARE project, once we have a candidate of promiscuous enzyme or a huge collection, then we ask the company, which is the product you want to convert? And then we make a second or third screening. And then under which condition you want to test? And then under this solvent, so under this temperature. And then from this pre-candidate, we come up with few candidates which are really of interest for an industrial point of view. And then together with the other partner, we express the enzyme in a cheap way. We mobilize the enzyme to make more robust enzyme in the process, and so on. And then this just last slide, I wanted to tell you. So my feeling is that in the companies, they normally like, or they normally choose at the first option the chemist. And then always they will think about the enzymatic process if the chemists fail. So if they don't find a way to produce biochemical methods, so then they will ask the enzyme. And then in the future, and all of you, I mean, you are the future of marine biodiversity or biotechnology in general. So in the future, we need to change this around. So I'm dreaming with the company which is thinking first in the enzymatic way, and if there is no enzyme that make the process, so then think about chemists. But this should be changed in the future. At least within Inmari, we have created one of the largest collection of enzymes, which so any company which approaches us, so we can test whether they can convert the product they are interested. So these enzymes are stable, active, and anti-selective, promiscuous, and so on. Okay. And for this, I want to conclude that I hope I have convinced you that biodiversity in the ocean provide a really nice source of novel enzymes, that if we complement cultivation metagenomics approaches with efficient expression systems together, we can come up with promiscuous biocatalysts that are able to convert different substrates under different conditions. Through the characterization of all these enzymes, we can do predictions. This is important. And I'm sure that the marine metagenomic biotechnology represents an important part of the third industrial revolution in the 21st century. And that's all. So if any questions, so then you can ask later on. Mm-hmm.
So we thank you very much for your seminars. They were very didactic and interesting for our students. We can see with the audience, which is really full here. So do you have any question, maybe from the audience? Our students might be a bit shy to ask in English, so you can ask in Spanish. Podéis preguntar en castellano y podemos traducir la pregunta if you wish, so we can translate it if you feel shy. Do we have a microphone or maybe? So.
James,
We,
no question?
yeah.
In some of the lakes, because actually we have this thalassohaline, which are saturated, let me say, seawater 10 times concentrated, or we have this magnesium-like, and they are somehow upper interface different, but not in majority of the, because we have always dominance of some proteobacteria in upper interface oxygenated. But if you will have, for example, or you are working with Medea, which is a sodium chloride concentrated lake, which has interface around more than 30 meters. It's really, really diffuse. Then you have this strange dominance of epsilon proteobacteria, which are actually already mentioned in Black Sea. They existing when the gradients is not so sharp. But in majority, yeah, because this system it's somehow it doesn't like to seawater microbial community at all, and all of them they originated from the this buried evaporites, and in general the community it's more similar between them than between seawater and the lake.
Can I say something? So there is a lot of, from enzyme point of view, there is a lot of discussion now about the ancestral enzymes. So how the ancestral enzyme from million years ago and the enzyme from now are different or not. So it seems to be that the ancestral enzymes are more promiscuous. And what is nice in this extreme environment that Misha commented is that these deep sea water lakes were created million years ago. 5-6 million years ago. So at least. So probably they contain microbes that didn't have contact with the rest of the planet in these 6 million years. So they contain enzymes that didn't make any change in them. So it could be nice to get these samples, to isolate the microbes and also the enzyme, because probably they are not only from an industrial point of view, but also from a basic point of view. So it's something that are microbes and enzymes that didn't change in million years ago, not comparing with the ones that are here on the soil, because they are just with the climate change, changing temperature, changing conditions, changing pollution, and so on. So it's nice to have this kind of facilities and sampling.
I may comment on that as well. So in one of the first metagenomic papers we ever published, which was based on the petroleum enrichment of that very Urania interface, it's the Mediterranean hypersaline anoxic lake. So we did lambda-phage library, and we looked for esterases, and we found one esterase. We found actually 5 groups of different ones. And exactly from that site, we found some protein, which we until now, we cannot find the affiliation in, you can imagine how many genomes are sequenced now and available in database. But there is one which we still fail to find in the database. So that was something really unique coming from beneath. Yeah? And we're still puzzled where it belongs, which organism. And it's a very bizarre one. It was very bizarre protein, yeah. So it was quite large, and it has few active sites, and it did a few different things depending on the pressure, depending on the salinity, and depending on presence or absence of oxygen. And till now, we still are puzzled where it comes from. Yeah.
Yeah, well, I'm not really the expert in bioinformatics. I'm more in the experimental part. We have within INMARE a different bioinformatics college. So you can apply, in general, to different kinds. I cannot tell you exactly which are the softwares and tools, but you have two different or a few different kind of methods. So the one to assemble the DNA sequence, because you sequence the DNA, you get pieces of DNA that then you need to assemble. So the complexity of the assembling process is higher, it's much higher the diversity is, because you will have pieces of DNA that are really similar. So there is a lot of bioinformatics expertise behind the assembling of the DNA to get the full DNA sequence, okay? And then you have bioinformatic tools just to predict enzyme functions, okay? To reconstruct the genome or the metagenome, but also to predict the function from the sequence. And then the most important thing also actually, and that is a partner here from Bremen, from the Max Planck Institute, is that every time you sequence a genome or metagenome, 40% of the proteins are hypothetical because we don't know what they are doing. So a lot of bioinformatics is being applied actually to predict the function of the gene, which actually we don't know. I cannot give you the detail because I'm not much expert, but in general these are, probably my colleague has a different opinion, but these are the different possibilities.
So we have Antonio, who is just on the rear seat over there. I know that he's using this network reconstruction, like coherence network reconstructions, to actually to reveal the function of you know, these hypotheticals, real ones. They're unknown, so to speak. So they appear in one organism, and then they're unique. They have no homologs. You also have conserved hypotheticals, where you can predict some general function, but sometimes they just form clusters, and you have still no idea what they do. In many cases, even you have the protein structure. In many cases, when you do naive expression and structural analysis, you cannot predict function. So there's still a lot of things to do. But I know that Antonio is a big expert in coherence networks. Okay.
So maybe you can approach Antonio later after the talk for more details. So is there any, so our speakers must go for lunch, so is there any last question that you would like to ask? No? May I try maybe for last question? So you've said through the project, in my project, you've generated a big collection of enzymes. And regarding the technical improvements about high throughput screening methods, have you advanced a little bit on this?
Yeah.
Or?
I think it was one of the major problems when the IMARE project started. At that time, most of the academic group was investing a lot of manual time, and then we invest a lot of time to create this high throughput system, not only to screen the enzymes, to sequence the DNA, but also to make high throughput cloning, high throughput expression, high throughput testing under environmental conditions and so on. But I think not only we created this collection and a lot of knowledge behind, but also a lot of robotic facilities. And because these robotic facilities, so then the number of tests increase exponentially. Yeah, I think this is critical, and then we are in a time where this technical development is really the best. I mean, there are a lot of companies producing a lot of robotics. I think we are in good time to use.
Thank you. So thank you very much to all the speakers, and maybe we can... Thank you for your time, and we see you now at the lunch.
Thank you.